Right. Okay, so it's my pleasure to introduce our speaker tonight, Heidi Gartner. Heidi is a deep sea biologist at Fisheries and Oceans Canada. In the past 15 years, Heidi has studied marine life from the intertidal to the ocean abyss, covering topics such as taxonomy, invasive species, and species at risk. You may recognize her from her past presentations in her previous position at the Royal BC Museum, where she was collections manager for the marine collections. So tonight she's going to cover the work of the Deep-Sea Ecology Program over the past 5 years. I'm looking forward to this because I know there are lots of neat photographs and deep sea pictures to see, maybe a few videos. So with that, I'll hand it over to Heidi.
Thank you, Phil. Sorry, I find it too bad that I can't often see people once I start my presentation. So I was just trying to sort that out. Thank you all so much for coming and for wanting to hear about our deep discoveries tonight. Yeah, my name's Heidi, and I'm sharing the work of the discoveries of the Deep-Sea Ecology Program at Fisheries and Oceans Canada. I feel a little bit silly presenting this because I've actually only been with the program for about a year and a half, a little under, and we're actually celebrating the 5-year anniversary of the program. It's pretty fantastic. I would like to acknowledge that Dr. Cherise Dupree and Tammy Norgard have been the main part of the program for the better part of these 5 years. And I'm going to see a lot of we've discovered or the program's discovered. It's actually them. It's not fully me most of the time. So very thankful for them and their work. And it's been a fun team to join. Cherise also likes to give, or I've adapted this from a similar talk that Cherise has given. And she likes to call it Deep Dark Secrets, except for now she calls it the not so big secrets because we're starting to figure things out. But really, what's fascinating about the deep sea work here happening off the BC coast is that a lot of this is still kind of frontier research. So this is Tammy and Cherise, and they've been the head of the programs for the Deep Sea Ecology team at Fisheries and Oceans Canada. Just to orientate us, this is, the bright red dot is where I have the privilege to live and work on the traditional territories of the Wasanic people. I find that a lot of times people use this orientation of the map to help orient where we are in the world. But for what I'm talking about today, this is actually a much more fitting view of where we are in the world. We're planet blue, where 70% of the world is ocean, not Earth. And it's incredible. We know more about the surface of the moon and now Mars than we do about our own planet, about the deep ocean. So only about 10% of the ocean is mapped or well studied. So what's really fascinating is that this program has only actually been in existence for 5 years. It started in 2017. There was research that happened in our deep sea, but we did not have like a program dedicated to this through Fisheries and Oceans Canada. And it's just important to think about why this has come about. A lot of our funding does come from marine conservation targets and the ideas of that. I'm sure a lot of people have been hearing about global international agreements to try and protect 25% of the ocean by 2025 and 30% by 2030. So we're thinking about that with this lens that in the deep ocean, less than 10% been well mapped or well studied, and here we are trying to protect a large part of it. So when I say deep ocean, what am I talking about? Well, in the world, the deepest point is the Mariana Trench. It's over 11 kilometers deep. What we're working with here in Pacific Canada, a bit shallower. So we have the majority of the offshore within what we are, within our economic exclusive zone. So what belongs technically to Canada or BC. The majority of that is actually at 2,500 meters depth. The Global ocean is about 3.5 kilometers, so just a little bit deeper, but we still have some very deep waters here off the coast of our BC. There is one part of our ocean where the tectonic plates are spreading apart, and there's a rift that is about, well, it's a little over 3.5 kilometers deep. And it's actually amazing because there's this tectonic plate action that is literally tearing apart like a huge super volcano called the Explorer Seamount. So deep ocean, that's what we study. Who lives there and how? Well, some of the trends in deep sea are kind of fascinating in the sense that they're not what you typically expect. So one of them is giants live in the deep sea. So there's a tendency for species of invertebrates and other animals to be larger than they would be if you were to look at their shallow water counters. You can tell I borrowed this part of the talk definitely from Cherie. So she's showing like respective to her size, some of these animals and how large they can get. Why is that? And there's kind of two scientific rules that kind of explain why some things are so much larger. And one is that here we are in this deep ocean, food is scarce. And larger animals tend to be more metabolically efficient than small animals. And so to become more efficient in a deep sea where you don't have much food, you become larger. The other thing is that a lot of times, well, there's low temperature in the deep sea. So it's often at the deepest depth, around 4 degrees, even colder in some spots. And large cells and longer lifespans are usually associated with these cold temperatures. And this leads to increased size. And what's a fun example to think about is isopods. These are the little pill bugs that you would see like when you go out on a hike and you disturb a log or something like that. They're just, you know, a few centimeters. They roll up into little balls. The deep sea equivalent of that is 50 centimeters big. Usually lives at around 2.5 kilometers depth. And it's really tough when you see image of it to really get a sense of how big this is in the deep sea. So this is an example of one from a museum. As you can see, it's over more than 2 like hands lengths. But a clever comparison is that it's about 25 times the size of you. So if this is regular you, this is deep sea you. Or even better, I think this helps put it in perspective for me, is that it'd be you standing at that famous statue, the Christ over Rio. Just imagine standing at the bottom of that and then thinking about your deep sea version would be that large. So it's a fascinating phenomenon that happens in the deep sea. The Hackeye magazine, I'm not sure how many of you know it. It's a local magazine that produces stories about all sorts of marine related topics, but from around the world. They recently shared about a study that was done on the genome of this animal. So they sequenced the genome and found that it's a really large genome, and that the sections that are really large are so-called jumping genes, which are things that would help be related to DNA mutations and would help the animal be more adapted to stress. And what's also fascinating is that they were able to determine that some of these mutations and these extra large parts of their genome were related to their thyroid and insulin production. That shows that, again, that might have an effect on how they're metabolizing their food. So this is an animal that lives in the deep sea where food is scarce, and when it eats, it can actually expand its stomach to fill 2/3 of its body. And then it has all these characteristics that are making it metabolize food slowly. So this is an animal that can eat once every couple of years, which is just fascinating to me. So this is interactive. If you'd like, some fun questions for you. So how big do you think those anglerfish are? If you want, you can share your answer in the chat, or you can just, think about it out loud. It's interesting. But I have to tell you, this is a bit of a trick question. After talking about deep sea gigantism, this doesn't happen with all animals. The anglerfish is misrepresented often in movies, for example, Finding Nemo. It's not that huge. It's more the size of actually the clownfish Nemo that's trying to find it. They're only about 2 to 18 centimeters big, the average. They can have ones that are larger, but they're still not the way they're perceived lots of times in the movies. So sorry for the first trick question. But it does lead into who else lives in the, or another characteristic of who else lives in the deep sea, and that's really highly specialized species. So the deep ocean is really cold. There's no sunlight. There's often really extreme environments as well in terms of like, think of all the pressure if you're that far down. And so a lot of animals come up with really interesting adaptations to living in these places. So for example, the anglerfish we just were talking about, it has this lure on the front of its head that has bacteria living in that lure that can do bioluminescence so that it can draw in prey and feed on the prey. This barrel eye fish, it has a tubular eyes and a transparent head. And it's one that lives almost in the mesopelagic, so just below the sunlight, but it looks up and it can see the outline and silhouettes of all the fish that are outlined by the last little rays of light coming down. And then that last but not least, this tripod fish has a crazy adaptations. Its pectoral fins and its tail fin, the rays of them have been adapted to be these long tripods, and it uses those to stay still. Again, you want to reduce your exercise for metabolism for scarce food. And then it orientates itself into like the current, and it will use its pectoral fins to help try and flat food towards its mouth. Like just absolutely weird and fascinating animals that adapt to these deep sea environments. Another trend in animals that live down here is that they are long lived. So again, this is partly related to the giants rule kind of thing, but it's also, if you think about these habitats, they don't go through such temporal or short daily dramatic seasonal different changes. These are usually things that change on more like geological time scales. They're not dealing with the fluctuations of the sun nearly as much and that sort of thing. So some of the animals I'm going to talk to you about, it's just amazing to think about this. So rockfish, particularly the rough-eye rockfish, is more than 200 years old. These are animals that live around our seamounts, and you know, it's just so hard when you're a human who, from the perspective, to just think, oh yeah, there's another commercial fish species swimming by or something like that. But this fish was born 7 generations ago, before my, you know, great grandparent was born, before Canada even existed, you know, or Canada has just been formed. The corals that we have on our coast, some of them have been, they measure how long it takes them to grow. And some of the really tall ones they suspect are over 300 years old. Again, 10 generations of our family. And then last but not least, we have some of the oldest known animals on the planet in the invertebrates of glass sponges. So glass sponge reefs are when an animal that makes its body entirely out of glass, it produces this gorgeous rigid structure. And when it dies, and if it doesn't get knocked over or damaged or something like that, the new one can settle on top of it. And so they create these incredible reefs, and they're known to be thousands and thousands of years old. I'm not sure if any of you have heard recently of the Netflix documentary called My Octopus Teacher, but this is your next kind of quiz question. She was a mother octopus, and at the end of the film, she guarded her eggs for 4 months. So again, chat or not, wherever you want to put it, even just if you yell out to yourselves, how long do you think a deep sea octopus would guard their eggs? So the thing with the deep sea is that, do you know what? It's actually hard to know these things. It's so hard to get down there to study these animals. So often in the past, what we learned about our deep sea was through bycatch, through fishing, that sort of thing. But there was this really unique instance with the deep sea octopus, Granulon boreopacifica. This is what it looks like. We see it when we do our surveys. It's a very cool octopus. Like all the octopuses, they show fascinating behaviors. They use their chromatophores on their skin to camouflage or to signal, or just incredibly complex animals with incredible behaviors. And the same species lives down near California, and they had an opportunity, the researchers had an opportunity.
Huh?
Are you sessioning?
It's not a session, it's a presentation.
Right.
Sorry, you're not muted. Anyway, the octopus, they had an instance where they were able to repeatedly come back to the same spot where they spotted one of these females guarding her eggs. And over time, they went back and forth, going out for surveys, and she guarded her eggs for over 53 months. No one had even expected it would be close to that. That's over 4 years. I don't know how much you know about octopus biology, but the females tend to their eggs without feeding, without moving very far for over 4 years. This was an absolutely fascinating discovery that was actually relatively recent. The last thing about deep sea animals and just some sort of trends for them in the deep sea is that our food webs are made up a lot more of detritivores and scavengers. So on this slide, you see, you know, the slightly more typical food web on land of, you know, solar energy gets absorbed by plants and it gets transferred into energy that works its way up a food chain. Although I think this is hilarious because Cherise thought that this looked like a hummingbird, and she thought it was a very random sample to use for a food web. And in the deep sea, it's similar. We have sunlight that gets processed by photoplankton, which then gets eaten by bigger things and works its way up the food web. But it's a bit different in the sense of it's way messier. And it's in this very interesting medium, and there's a lot of waste. So what happens in the deep sea is the phytoplankton, not all of them get eaten. So some of them decay and fall into the water column. They die. The things that do eat them have excrement, and then they die as well. Not all of them get eaten. And so actually a lot of the deep sea is about the kind of waste products of the upper ocean falling down to the deep. It does get entrained with things like microbials, and globally about 3% of the carbon produced at the surface sinks down to the deep sea. What's amazing is this 3% supports an incredible diversity of life down in the depths. This stuff that falls, the waste products, the pretty name that we use in deep sea biology is marine snow. And if you ever tune into, we sometimes live stream our expeditions, you will see that, like, you can actually see almost like fuzzy stuff falling in between the camera as we're going down. So with all this snow falling down, marine snow falling down, this is what we typically used to think the deep sea looked like: a bunch of mud, just flat mud, not a lot of life. But it does go to highlight that a lot of the low flow and flat environments have kind of more of the detritivores, the things that are cleaning up the bottom, or scavengers, things that find dead things. And what's fascinating is once you add a little bit of current, or a little bit of relief that makes current, we move into this other style of feeding called a filter feeder. So it's almost more like a sieve. These animals put their bodies up into the water column and are able to filter food from this marine snow falling down, creating incredible habitats. Lastly, there are other sources of falls or deaths to the ocean, and they do create really spotty, unique environments down in the deep sea that do support incredible diversities of life. And if you pay attention there, there's that Dennis, the deep sea isopods are really specialized at taking over carcasses. And that crocodile, I believe, was like a forensic experiment, basically, that was studied in the deep sea. We did something similar with pigs on the west coast of Canada. So I will touch on this a little bit different. There's also, or a little bit later, sorry, but there's also another different type of animal called chemosynthetic animals. I'll highlight them in a moment, but it's just a whole other story that's just blown people out of the water. I know, I just said blown out of the water, sorry. So now we live down there. Why do we care? Well, I've been talking about the lens of the deep sea and conservation. So what does that mean? So we talk a lot in my world about EBSAs, ecologically and biologically significant areas. It must meet one of the 7 criteria that follows after this. It has been usually designated by a formal governing body. For example, here in Canada, it's Fisheries and Oceans Canada through the Government of Canada. But it is a United Nations Convention on Biological Diversity standard that is shared around the world. And so to be an ecologically or biologically significant area, you have to score high on one of these 7 features, which is you have to be unique or rare, provide special importance for life history stages of species, be important for threatened or endangered species or declining habitats, be vulnerable, fragile, sensitive, or slow to recover, have really high biological productivity, have high biological diversity, or naturalness. So are there EBSs in the deep sea? Yes, there are. We are so unique. We have an amazing amount of diversity in our deep sea. The first one that's considered an EBSA is called seamounts. These are structures that rise over 1,000 meters from the base to their summit. They are literally underwater mountains. I'm going to actually talk about them the most last. They've been the focus of a lot of study here in BC. We also have hydrothermal vents. So hydrothermal vents are EBSAs. They're ranked high on almost all 7 of those points. And hydrothermal vents are these fascinating environments where superheated water and toxic chemicals actually support hotspots of life. I know it doesn't sound right, but essentially what happens where seafloors are spreading or there's cracks in the mantle, if we think back to the good old tectonic plates we learned in high school, or water starts seeping in between these cracks and it goes down, the seawater, cold seawater goes down and it gets superheated by the magma. This magma, as it gets superheated, it gets superheats the water and it gets really pressurized and it starts shooting back up through the crust. And the superheated water picks up a lot of minerals as it goes up. And then the superheated, highly rich mineral water gets shot out of the crust and it hits that, again, cold seawater, and it precipitates out metals. These metals also can provide hard structures, creating chimneys and high reliefs. And I've just described a highly toxic, really hot, up to 400 degrees Celsius water, very strange environment that has been discovered to be a biodiversity hotspot. And it's all based on this crazy idea that instead of sunlight, life can exist based on chemicals only. In these deep sea environments, they found a wealth of life that exists around them, either through bacterial mats or bacteria that are symbionts with things like tube worms, clams, that sort of thing. And they extract the sulfur, the metals from the seawater, from these hot environments, and then they produce food for the animals that they're living in. And this discovery really changed how we understand life in general. All life before this discovery was thought to be based on photosynthesis. And what's amazing is this deep sea discovery was only in 1977. So that's, around 44, 45 years ago. It's relatively recent. And no one knew that these habitats existed or that life would be so bountiful there. What's a very cool story for the west coast of BC is that we have Dr. Verena Tunnicliffe. She was a researcher at the University of Victoria, and she discovered the first ones in Canadian waters in the early 80s. She went on to study these habitats. We have a bunch of hydrothermal vents in Canadian waters, and she went on to discover over 80 new species to science. And then on top of that, so many people were inspired by her that another 10 deep sea species were named after her. Our program has largely been compiling information from past research and using it to inform spatial management, but we are incorporating some research on them into our expeditions that we plan every year. And for example, this is an image of this year. We went to West Valley hydrothermal vent site, and it hadn't only been briefly described through very brief imagery like 30 years ago, and we went back to look and see what the habitat looks like and if it's still active or not. And no one had ever set eyes on this 35 years later. And this does a really good job of showing that even though we didn't see active venting, we saw the historic remnants of an old active vent. So this chimney structure that you're seeing built up here, the hard structures here, when you add this sort of relief to what would typically be a relatively flat environment, you get incredible biodiversity starting to bring up, because it brings up extra currents. Life can grow on it. And as you can see, it starts to support a whole other ecosystem. So where are scientists most excited to look for chemosynthetic animals next? Again, a fun question for you. I feel like all of these are possible. Maybe the microwave's questionable, but really the most fascinating thing is this changed the fundamental premises of how we understand that life can exist, including anywhere in our universe. And so scientists have been really fascinated by these discoveries, that life can be based on chemicals alone. And they're looking to other places on our planet. And wouldn't it be fascinating if life on other planets doesn't look like typical aliens, but more like these strange tube worms that live at hydrothermal vents? So just super cool. Another chemosynthetic environment we have are called cold seeps. They're the opposite of these hydrothermal super hot vents. They're more like slow, bubbly methane seeps. They're not super hot, but they do the same sort of thing. We have bubbles that come up out of the Earth's crust with these methane-rich bubbles. Bacteria form around them. They start to form hard structures. And again, it has the same cascading effects on the environment. We have the symbiotic relationships with bacteria and some animals, and then other animals start to colonize the hard places generated by these abscesses. Along our coast, we do have cold methane seeps. It has not been a main focus of the program over the past few years, but we did go to a site, and what we're testing is that there's new information and new ways to map where methane seeps are. And so we've been working with our partners who would largely capture that information about where the methane seeps would be, and then going and ground truthing it with our cameras to see if we can find, these bubble signatures they get in their sonar. And this was a really cool site because we didn't actually see the bubbles, but we saw the evidence of a historic cold seep environment. So we'll be expanding this in the years to come, finding and mapping more, and then considering their protection as well. There's also coral and sponges. They meet one or more EBSA criteria, but on a case basis. As animals without backbones, corals and sponges are very fascinating because to a typical person, they might not be so interesting in the sense that they don't have eyes, they don't have centralized brains, they don't look like a typical animal, but they provide incredible services in the deep seas. They provide habitat heterogeneity. They grow up into the water column, providing extra habitats for things. You often find that they're nursery grounds for other animals, commercial species, endangered species. They provide microhabitats for little things to fit into. And what's amazing is in BC, we have glass sponge reefs. They knew about glass sponge reefs from the fossil record, but they hadn't seen any in the world until 1987, and the first glass sponge reefs were discovered. They have since been protected in many parts of BC, and we're still finding more of them. And then what's even more incredible is that just last year, in 2021, in partnership with the Central Coast Indigenous Resource Alliance, particularly the Kitasoo and Hehe Nation, that they discovered the first coral reefs in Canada. This is actually a big discovery in the sense that this coral is known in other parts of the world, but was not suspected to be able to be here in Canada based on our conditions. And we found it through working with our Indigenous partners in a very narrow band in the Central Coast. And this is a huge discovery, a healthy reef that no one knew about ever existing. And I'll just show you very quickly kind of what this imagery looks like when we go down and see new life. I think they're incredibly beautiful, and on top of providing incredible services. Oops. And I hope I'm getting across that even as a newcomer to deep sea biology and being a biologist in this world, it's fascinating to me how much we are just discovering, how much is new and exciting, and how much there is to do. So the last habitat that's an EBSA in BC is seamounts. So these seamounts are underwater volcanoes that literally rise over 1,000 meters in height. They again are EBSA ranked high or medium for all the criteria, all 7 criteria they had. For example, some of our bigger seamounts are actually bigger than Mount Baker. What we see visible from, Victoria, Sydney, like you often see it in the distance, the snow-capped mountain. We have mountains like that underneath the ocean in what was, originally typically thought as this flat, muddy habitat. And as you can imagine, these seamounts are incredible hotspots for life. They, first of all, ocean currents hit them and move up them, creating, extra flow, extra current, bringing more of that marine snow and available food onto it. They provide hard structures through their magma that's spilled out and the sides of the mountains for things to settle on and grow. Once they grow, other animals come in and live amongst them. And what's amazing is that they've determined that each seamountain actually should be defined as having a 30-kilometer EBSA around it, because these cascading effects go out so far that they found increased biodiversity of all sorts of marine life, cascading up to like the big predators, even migratory birds, up different fish food chains. So we have all this in BC. Why is the deep sea off of BC so remarkable? Well, the idea being that the more niches you have, the higher biodiversity you have. And what was really unique to BC is that we have really active tectonic activity in our waters. So we have 3 tectonic plates that are within this EEZ, or this economic exclusive zone, that is in Canadian waters. These create, these massive underwater features like these volcanoes or these hydrothermal vents, and, the extra complex habitats that are associated with them, everything from like the little complexity the lavas add. So we've already inherently got more different habitats in our waters. And then, as well as when you go through the water column, BC has very strong oxygen zonation. We have a very strong an oxygen minimum zone in our coastal waters, which creates different habitat tolerances, and again creates different habitats that different animals can live in. So we are actually here in BC home to Canada's only seamounts. There's no seamounts anywhere else in Canada. There's no hydrothermal vents. There's no cold seeps. Only here in BC do we have any of those features. And these are all known, again, as EBSAs, like really important ecologically and biologically significant areas. We are the only place in the world with glass sponge reefs. And we have a globally high, unique density of hydrothermal vents in deep sea seamounts. Again, that's in the protection of a government. It's very rare for this to be within a country's jurisdiction. Why should we care? There's so many reasons we should care. Everything from food biodiversity, all the inherent things, but the deep sea does a lot of services for humans as well as that intrinsically to protect our world. Everything from, it actually is one of our big climate change regulators, to it actually might present resilience for species. We're writing a paper up about how one of our seamounts might have provided a refuge from the sea star wasting disease for some sea stars. Oops, sorry, just heard someone. And so it's just a remarkable place that we have off the BC coast. And it's kind of not a surprise because we actually, that there's so many amazing tools and important research going on in this field. So actually BC was the home of Pisces 4, one of the earlier manned submersibles. It was the world's first undersea cabled observatory, known as Venus, and then it became Neptune, and now it's Oceans Network Canada. We actually have one of the world's best scientific ROVs, or remotely operated vehicles here, the ROPOS. We now have the deep sea research programs at Fisheries and Oceans, but there's also work going on at the university and at Banfield. They have their ROV. And we are actually the world's first marine protected area for hydrothermal vents. They were established in 2003. So this program that I'm talking about at Fisheries and Oceans Canada, it was largely made with the idea of conservation. So now we know we have, or how do we know what to protect in the deep sea? And so the first question we kind of had is, what's there and where is it in our coast? And so the first thing this program did was look at where the hydrothermal vents are and the seamounts. And that's what's visible here on this map. Oh, someone's not muted and you just screamed. If you wouldn't mind muting, thank you. There was a report just before this program was built that determined there was 20 seamounts in the offshore Pacific. And in the 5 years of this program, we are now up to 65 seamounts. So if that doesn't help hit you how little we know about our oceans, that since 2017, we've added 45 new seamounts, things that are almost the size of Baker, we didn't even know was there. Absolutely fascinating. So what originally happened is Cherise did a lot of this through modeling and then ground truthed it with sonar and then reran the model, and we're working it out there. So we haven't confirmed every single one of these, but when we go out to confirm them, we actually find more because the models actually underestimate summit heights usually. So just to show you what this looks like, what it looks like to go out and map is on the vessels, we use eco sounders. So it's that same idea that bats put out a sound, it pings off something hard and it comes back. So that's the same idea with sounders or eco sounders. And what it looks like here on the left on the screen is a multi-beam echo sounder. This is the gold standard. This is what we would love to do in BC. It means that there's multiple sounds going out, so you get broader swaths and more details. On the right, we can also do it with a split beam echo sounder. It's just one noise that comes back out, so you kind of get the relief, but you don't get the full picture. And what's fascinating to me on the left there is that Union Seamount, the picture of this, was one of the most fished seamounts in all of our waters. And we did not know what it looked like until 2022. This summer was when it was first mapped with a multi-beam echo sounder. And then on the right, the split beam, this is another case of where we went to ground truth models and found out there was actually 2 seamounts where there's supposed to be 1. So when I say we go out and do this research, yes, a lot of the work is relying on science that has been done in the past, but we're also trying to do our own science as well to inform these conservation targets, that sort of thing. So I'm going to show you an example video from the expedition that happened this summer. That was 2022. The program went out 4 of the 5 years. Obviously, 2020 was complicated for everyone and we were not able to go out. in those 4 years, you'll see a highlight. This is just a highlight, but usually these expeditions to do research are multidisciplinary. So we go out and we look at oceanography. We look at mapping, doing this mapping. We try and get underwater robots under to have a look in what's down there. But what's really fascinating is this mapping the ocean that we were talking about. There's no multi-beam echo sounders on any Coast Guard ships on the Pacific coast, which is just mind-boggling in this day and age. And so we wanted one this year, and we got some sort of deal where we got a multi-beam echo sounder, but it wasn't hull mounted. So someone was like, well, you're gonna have to build a pole. And we're like, okay, sure. I had no idea. I'm not a specialist in this area. And when I saw the size of the echo sounder, and when I saw what they meant by building the pole, in this video, you'll see one woman she was from Ocean Networks Canada, built this incredible electromagnetic system to put this multi-beam over the side of the ship. It was mind-boggling. So this is what a science expedition looks like.
The Deep-Sea Diversity Expedition has returned from conducting science to support the multiple nations co-creating and co-governing marine conservation areas off of the Pacific coast of Canada. We explored new and long-term monitoring habitats off of Vancouver Island and Haida Gwaii. Our team was the first to lay eyes on Sept 5494 seamount near Haida Gwaii, the closest to shore seamount and the only one in a newly proposed conservation area. We returned to Skong-Kinglas-Bowie Marine Protected Area to resurvey long-term monitoring sites, and remarkably were able to visually detect some ecological changes over the last 4 years. Haida scientist Gil Jada led the dive to the ecologically and culturally significant peak of the Skong-Kinglas-Bowie seamount, known to have been a Haida island when sea levels were lower. The dive to the summit was awe-inspiring. The shallow, sunlit cliffs and peaks gave rise to towers of thousands of rockfish, visible in both the submersible camera and the scientific echo sounder. A diverse taxonomic collection of 100 specimens from a dozen phyla could possibly offer new species to science. A priority of the mission was to widely communicate the science to a local and global audience. Events were jointly hosted by communities, partners, and the at-sea science team, reaching the general public, scientists, and over 40 classrooms around the world. The live streaming of the deep-sea footage and chats had a worldwide reach and allowed us to interact and connect with viewers and gain real-time input from shore-based collaborators. Using the ship's echo sounder, we profiled and confirmed 8 seamounts, 4 of which are newly discovered. The ship was equipped with a multi-beam echo sounder, which allowed us to gain high-resolution seafloor data, including that of Union Seamount, the second tallest seamount in Canada, which had been previously unmapped. A broad group of determined collaborators came together to equip our vessel with a one-of-a-kind mounting system for the multi-beam to enable our ambitious project to map the deep. We added to the global array of Argo floats that autonomously measure chemical and physical properties of the water column for 5 years after deployment. Continuation of oceanographic sampling in the offshore area added important information on the chemical and biological properties of the water column in seldomly visited areas. We explored the seafloor of future conservation areas for chemosynthetic ecosystems, home to endemic animals, including being the first to return to the West Valley hydrothermal vents 36 years after they were discovered. This science was a United Nations Ocean Decade endorsed activity and will contribute to our global understanding of the deep through the Challenger 150 and Seabed 2030 initiatives.
So I realized the time. I'll try and wrap up in the next 5 minutes. Does that sound appropriate, Phil? Or would I do have a little bit longer to go?
Take as much time as you want. If people have to go,
okay.
They'll leave.
Okay. Yeah. So these expeditions are incredible. They try to tackle a lot of different aspects of finding out what's going on in our offshore. We do partner with the Council of the Haida Nation, largely especially in San Kingless Bowie MPA. And with the New Challenge Nations has also been a really active partner of our expeditions as well. Essentially, what I hope you kind of get a sense of from this is that seamounts are absolute incredibly biodiversity hotspots. They have the corals and sponges, they have fish, it has these cascading effects. We've got these really long-lived animals. We have animals that start to somehow find this productivity and incorporate them into their migratory routes. And everything too from just opportunistic sightings. So for example, at the bottom of this page, you can see a shark scratching its body along a log. That's a behavior that had never been documented before. And we've now seen it twice of sharks using anthropogenic debris to scratch their bodies. Just incredible. And sometimes it's not just about the animals, it's about like just even putting cameras under the water. You get to see things that you would have never caught from traditional methods like trawling and fishing and different ways that we used to study our ocean. So for example, on the left hand here of this slide, we have, we discovered on a new seamount that we didn't know about before the past 5 years, that it's a nursery ground for a new species of skate called the Pacific white-sided skate, or the Pacific white skate. I know its scientific name better than its common name, sorry. So what you're seeing is these little purses here. The gold ones are new egg cases and the black ones are old ones. So these animals are coming up from depths, laying their eggs on the summits of these seamounts and going back down, which is just fascinating. And when we send these cameras down, we take transects, we take imagery. Yes, we're trying to find out as much as we can in time, but one of the complex things about studying this is that, you know, we collect 2 weeks worth of scientific data and then we spend the next 5 to 10 to whatever years analyzing it. So we're trying to describe these species. We're trying to then annotate the videos to try and get estimates of where biodiversity hotspots are even within these small things. So if you ever want to look up on iNaturalist, we have a list of all the animals that we're finding down there. It's amazing to watch the discussions because sometimes these things have never been found before. And then this is what happens in our annotation program, Beagle, is that every little dot gets annotated and that gets turned into spatial data for us about where we can find things. And we have sensors on the robots that we send down that tell us the temperature, they tell us the oxygen, they tell us all that fun stuff. We've also 2 out of the years had the opportunity to have one of these robots go down called an ROV, remotely operated vehicle, and they've got hands essentially, so they can collect things. So we were able to collect these animals. So this helps answer our annotators' questions of, you know, which species are they, what animals are they? And these were all donated to the Royal BC Museum. So we did that in 2018, and then we did it again this summer in 2022. They're not yet at the museum, but they will be soon. And this is what they look like when they come up and they get sent to the museum. And why do we send them to the museum? Well, we ourselves do whatever taxonomic work we can. We have Merlin Best, who is our taxonomic expert in corals and sponges. They do a great job, but we're not experts in everything. For example, Phil is a emeritus curator there, and he still sometimes works on the echinoderms for us to help us discover what some of these deep sea animals are. The urchin at the top is very fascinating. It almost has like hooves on the bottoms of its spines. I was watching one of these dives back when I was at the museum. And I said, hey, we've never seen that urchin before. We don't have it in our collection at the museum. I was able to communicate to them on the vessel. They were able to collect it, bring it back. And the world expert asked me to mail it to him because he'd not had more than one specimen ever in real life. And so just fascinating things like that. So then we can put names on things. The late Dr. Henry Reiswig was also at the museum, and he was the world's renowned expert in glass sponges. And he takes these animals that build their bodies out of glass, and he breaks down the elements all the way down to the little spicules. And these different shapes tell you what animal it is. And that's how he can tell the species apart, is each spicule will have different shapes. And this is really valuable for us. In 2018, from the samples that they pulled up, they discovered 6 new species of glass sponge, 7 regular sponges, 3 corals, and a parasitic zoanthid. Like, these are animals that no one has ever seen from science before. We also had new depth ranges and new range expansions for species. And then it's also just very valuable for our annotators. This was a new species of sponge that no one had ever seen before. Henry described it, but he also confirmed that these 2 slightly different morphs were the same species, just growing up, and one's younger, and when they get older, they seem to trumpet out into the trumpet section versus the little kind of bugle. So what this all leads to is we're DFO science, and we try and inform the people who, you know, make the decisions about protections and policies and that sort of thing. So they kind of asked Therese and Tammy, okay, now you've got all these seamounts, you've got all these amazing deep sea habitats. How do we know what to study and where? And so Tammy and Therese have taken what they can from the data, and they've started to provide information like, well, there are some generic trends that are starting to pop out. Again, it's really hard. We've only seen 19 of the 65 seamounts with any sort of imagery. But, you know, through this oxygen striations that we have through the water column, there do appear to be certain types of life that start to grow up. So for example, algae in the photic zone, then you get some filter feeders as it starts to become a little less oxic. Brittle stars and corals and sponges, they do really well as it starts to get hypoxic. And then when it's severely hypoxic, there's only corals and sponges. And then they wanted to know, well, can we classify them? Like, are certain seamounts more like other seamounts? And actually the best way to describe this was to create a map that essentially said, how much of this marine stow, also known as export productivity, all the stuff falling from the surface, how much of that is getting to a seamount? And then how much of a seamount, how deep is it? And what is the oxygen? And it ended up coming with this classification of seamounts that we can then map out, and then we know spatially where these are. It helps inform us how unique some of these seamounts are, which ones to study. And then Therese actually added an element where on this y-axis here, she scored essentially how much data these seamounts have on them, either from satellite imagery, from actual cameras going down. And then she scored anticipated changes over for these seamounts. This is related to climate change, change in fishing, other elements, and it helped direct that if managers want to see where changes are going to be detectable on seamounts, here's some seamounts to focus on. And also here's some seamounts that might go through the most large changes, but haven't got enough data yet that we should maybe be focusing on. San Kingless Bowie seamount is a seamount in our waters. And here's another question for you. I'm sorry, I'm running out of time, so I'll speed through this one. But the answer to this is it's all of the above. This marine protected area is incredible. San Kingless Bowie was once an island. It is known to have been visited by the Haida. There's oral traditions about a very neat story about paddling out there. Actually, when it erupted, apparently, because it was originally, you know, this underwater volcano, there's oral traditions all along the coast of coastal indigenous knowing of this rupturing. It's 180 kilometers offshore, and the main seamount, the supernatural being looking outwards, San Kingless Bowie, goes from over 3,000 meter depths to just within 24 meters of the surface. Like, that's in the photic zone with algae, and it's just an incredible place, and it has really big, significant, and biological implications. This is what it looks like mapped. Again, this has happened in the recent. And one of the big projects that we have in our program is that this is a marine protected area. It's already established. How can we monitor that the protections in place are being effective? And so this is one of the things that I went through, and I don't expect you to read this squiggly map, but essentially it's important to know that there's indicators that we've selected that might help us inform an indicator group needs. There's tools that we can use to look at them, and there's some monitoring strategies that would be effective. One of them is that we're looking at establishing monitoring area zones, actually. So this was a pilot study on Delwood Seamount, where we took really, really, really high resolution photographs from 10 meter by 10 meter swath. And then we built these 3D models. So this was largely processed by Georgia Clyde, one of our team members. It's also contributing to Lindsay Clark's master's project. She's at UVic. But what we can do is then visit these places again at time. And if you can see the incredible diverse detail at which we can look at one tiny little swath on a seamount. And we looked at an even smaller section of it. And what's fascinating is we went back from 2018 and 2021. We had different camera systems, so they're not quite perfect, but we were able to find the exact same marker 3 years later and look at the animals and look at the changes in growth. And to give you an idea of how difficult this is, the second camera system we used is called Boots. It's actually a drop camera. It's just towed behind a vessel. And the marker that we use for these long-term monitoring sites are the size of Frisbees. So we found a Frisbee on a mountain the size of Mount Baker at night with no lights, with a camera dangling from a, like, it's just fascinating if you think about it in the terms of what this would look like on land science. But they were able to look at like individuals 3 years apart. So, you know, at this specific site, again, it had only been 3 years, but no catastrophic events. Individuals hadn't changed in shape. We were able to get the environmental measures. And then it's interesting, we actually did it this summer in San Carlos Bowie, and we actually found more differences. So some sites were the same, where they didn't have much change over the 4 years, but some sites did have noticeable changes and catastrophic events. So really fascinating to be pioneering that work. Phil, I'm gonna check in with you again. That's the main part of like sciencey. There's a little bit about like deep sea impacts. Do you want me to take questions, or should I plug through with the last 5 minutes? I apologize. I realize I've gone over time.
Well, as most people are still here, we still have 42 participants. Sure, why not? Go ahead.
Okay, thanks everybody. I appreciate your patience. So what are we doing to the deep sea? I'm gonna go through it really quick. Pollution. Yes, every time we go down 2 kilometers into the ocean, we find like garbage bags and cuts and random stuff that we should not be finding. There's also pollution effects from things like oil spills, there's toxins, there's noise. What's really fascinating is not even current fishing, but one of the seamounts, we know the fishing had been finished for 30 years, but we found a net, a ghost net basically, and Charisse calculated essentially, you know, how many dead fish was on in the 5-minute window that we saw this net, and back calculated 30 years. And this ghost net has killed probably 1 million fish in that time. Like it was, I can't remember the exact number, but it's in that paper. Climate change is a really big one. Like we were talking about in our waters, we have this really already strict oxygenation zones. And what they've been finding, Charisse and a colleague, Dr. Tatiana Ross and Debbie Ianson, sorry, both of them from iOS here, that we've already are seeing a 15% decrease in our oxygen concentration. Our oxygen minimums down boundary is like expanding by 3 meters a year, mostly down, and our oceans are already getting more acidic. And again, if you keep in mind that we've got these deep sea animals that are used to things not changing on this kind of timeline, it's crazy. And then climate change has also had some really weird ones where, for example, there was 1 year where pyrosomes were really big off our coast, and those fell down to the deep sea, and just a completely different diet from what these animals are used to. So that was documented. And then the big thing is resource extraction right now. It's a really hot topic. If you haven't heard about it, please look it up. This is going to be catastrophic for the deep sea. Essentially, a lot of metal companies, a lot of companies looking to profit are trying to say, you know, all these smartphones, all the smart cars, all this clean energy, the only way to get metals for this is to ethically do it by going into the deep sea. That's not the only way to do it. So in our deep sea, we have polymetallic nodules In these blue zones, we have highly sulfide areas where these hydrothermal vents are. We have cobalt-rich crusts. This, for example, is one of the machines that they're planning on using to mine the deep sea. There's a human down there in the right-hand corner by the wheel for just perspective. And what a lot of scientists are calling out, or sorry, this is some of the mining, how the mining will happen. One of the big things is, yes, a lot of people are realizing we're going to directly disturb or destroy the habitat that's being mined. But there's this additional concern by the main way that they're going to process these is on these vessels where they'll have a really large dewatering plume partway through the water column as well. There'll be noise. This is all not yet in practice, but very soon. And so scientists are calling for a stop about this. We know next to nothing about the deep sea. What we do know about it is that it is providing incredible services to humans in all the very many ways. And actually what's fascinating is some of these companies that rely on smart technologies, that rely on these metals are like, no, even we aren't pushing for this. Like, we have engineers for a reason. We want to slow down until we at least find out. Let's look more into it. It's fascinating. Microsoft's behind it, BMW, Google, like some really big companies are being like, well, we're not actually the ones pushing for this. But in 2021, there was this deep sea mining got fast-tracked. There's an international seabed authority that regulates mining in the open ocean because a lot of these do not fall in economic zones that can be protected by countries. And this little country of Nauru is actually supported by not a Canadian company, but a Canadian-owned company. So this has very big ethical complications for Canada. They triggered what's called the 2-year cycle. So the ISA has 2 years to set up regulations about how to safely mine the deep sea. These were already in process, but by triggering this clause, they only have 2 years before they can go ahead without them if they don't produce them. And then some very strange things happening with the International Seabed Authority include somehow they just deprived the first testing of deep sea mining equipment without consulting all parties. So again, just looking at mining is bad. Very scary. There is some good news because we want to end on a good note. And one of them is that in 2000, so just 22 years ago, less than 1.7% of the global ocean was protected. We have since had the ACHI targets. The new target is to protect 30% by 2030. The great news is Canada is on track to meet these goals. This is largely by also adopting the United Nations Declaration on the Rights of Indigenous Peoples and going into some co-governing management of these areas. We are taking an ecosystem management based approach to some of this, and it is going to lead to a lot of marine protected areas. MPAs can't do everything. That's going to be the one complicated thing. But the 4 key things that they immediately, once you establish a marine protected area and or MPA, is that you do limit oil and gas activity, mining, and any dumping or deep sea bottom contact fishing. This is roughly what protections look like right now in Canada. The red one up on the top is Sanquilis Buoy off of the coast of Haida. The blue area with seamounts and vents is it's a proposed area, but that has not yet become a marine protected area. And that is largely the part of what we do at DFO, Fisheries and Oceans. So we are supposed to discover and explore the area, find out who lives there, determine where EPSAs are, define area of interest that then become or could potentially become marine protected areas. We can play over ecological overviews of them. We can conduct ecosystem risk assessments to determine, what are the main threats, which animals are most endangered. We develop marine protected area management plans. So Sanquilis Buoy Seamount is a marine protected area. It does have a management plan, and we are now moving into a monitoring framework. So how are we going to look at the success of this marine protected area over time at meeting ecological objectives? The very good news in the context of the deep sea is with all these planned protective measures in place, 81% of our seamounts will be protected, 100% of our hydrothermal vents will be protected. We're not sure about cold seeps. That's a big target of the program over the next 5 to 10 years to reach these targets. And corals and sponges, there's many different various protections. For example, there's now some in Bowie, or not Bowie, sorry, can't think of that. Anyway, we have several marine protected areas. There's a planned Northern Shelf bioregion network that's going to be there. And then, oh, in Howe Sound, there's also some coral sponge reefs that are under various states of protection. We have been using this data that we've been gathering to provide specific information about fascinating habitats on the seamounts. I can't tell you, I won't go into detail now because of time, but essentially sometimes we get down there and it looks more like a Dr. Seuss book than we'd imagine. So even though there's these patterns that we're seeing, just get down there and there's just some fascinating life. And to think that we've only seen 19 of our 65 seamounts, we've seen what we can light up with 2 meters of lighting with our robots. We only have, 12 hours in a day. We've probably surveyed less than 1% of this, and we're already finding just absolutely fascinating habitats and life down there. We work with coastal Indigenous partners, and it's really making this work even more fantastic, enriching it. We've had knowledge co-creation. So for example, we're working on a paper with our New Chalmette our partners and on about these observations of seeing sharks scratching up against anthropogenic floating material. And our New Chalmette co-author also thought that another way of knowledge co-production would be through traditional art. And for example, he's added like the gorgeous imagery of the copepods on the shark, like it's just amazing. With the Central Coast Indigenous Resource Alliance, they've identified some really important hotspots along the Central Coast for corals and sponges. We work on the Song Kingless Bowie Seamount Marine Protected Area. It's a co-managed marine protected area with the Haida Nation. Whenever we go on our expeditions now, we partner with nations, and we've had some incredible outreach to schools. That's just been fantastic. And last but not least, we just submitted the final kind of document summarizing all these 40-some features that have been discovered, and names of places are so important and they anchor us to space and time. And anyway, we are working with 17 coastal First Nations to actually name these features. So just incredible work there. And if you want to learn more, there's lots of papers. Best name to search is probably Sharice Dupree, but again, a lot of them are also DFO reports to inform management. We have an iNaturalist page. When we do go out on these expeditions, they typically happen every year. Look for live streaming, usually on Ocean Network Canada live streaming pages. You can actually go and find all our old dives on there if you ever wanted to just sit and watch. We do social media where we usually tag the year. We're actually on Shark Week if you want to try and find those scratching videos. And again, there's some really important research going on in the UVic labs with professors John Dower, Amanda Bates, and Kim Juniper. And now their graduate students, Lindsay Kark, Megan Davies, Daniel Labbe, and Pandora Gibb. And in addition to all of our Indigenous partners, all our wonderful grad students, the Royal BC Museum, the Coast Guard, there's a lot of partnerships that happen here. Ocean Network Canada supports the work, and many more collaborators at various universities. So thank you all so much for your time. I appreciate it. I'm sorry I had to rush through the end. I'm happy to stick around for questions, but if anyone else wants to go, there's my email. I would be happy to answer them. And if I can't answer them, I'll pass it on to Sharice. So thank you so much for your time.
Great, thank you very much, Heidi.
Yeah.
Very comprehensive coverage of your activities.
Yeah, I realized 5 years was a lot to squeeze in.
Yeah. If anybody has questions, you could put the question in the chat, or you can unmute yourself and ask Heidi directly. I was, you mentioned the first coral reef. Where was that again? I think I missed the location?
That's okay. I don't think I gave the exact location. Yeah, it's up in the Central Coast.
Okay.
Yeah, off of one of the islands. And it's been found in such a narrow band of environmental conditions. It was thought that our waters would maybe be too acidic to support the coral reefs over time, but they're now trying to extrapolate that and map that to like where the other possible habitat could be. Mm-hmm.
So it's in the channel between the outer islands and the inner Central Coast? Yeah.
Yes, if I can look up, I believe it was, I can't remember off the top of my head which island it was.
Well, maybe we need to keep it a secret anyway.
No, it's been out there in the news stories. It's everyone's welcome to look it up. Yeah.
Okay.
I'm just having one of those moments where I can't remember off the top of my head.
Let's see. We've got comments in the chat here.
Let's see. I haven't brought mine up. Sorry about that.
Thank you for a most fascinating and informative presentation.
Oh, thank you, everybody. Awesome. Thanks, everyone. I really hope that you all got my sense of like, I myself am shocked about everything. So I love sharing it with other people.
Yeah.
Yeah.
Amazing presentation from Kathleen Wilkinson. Amalis Riera. Enjoyed the talk. Thank you. Judith McNinch. Thank you for a wonderful presentation.
Oh, thank you, everyone.
Evie Chauncey. Thank you. Excellent. Pierre Lambert. Exceptional presentation. So informative. Thank you. Veronica Goidanich. Thanks very much. Amazing presentation. And Neil. Thanks, Heidi. Most interesting.
Thank you, everybody.
So I know I've in the past have been out on some of the ships way back when. I see the Tully is still going. It looks like it's getting a bit long in the tooth. Any chance they're going to build a new one?
Yeah, I don't know. Yes, they are going to build a new one. When it's going to come to the Pacific coast, I'm not sure. But one of the features that we had a opportunity to provide feedback as scientists is that we do want that multi-beam that you know, so many other vessels have, that we don't yet have that capability on the coast. So yeah.
So it sounds like everybody's learned a lot tonight and don't have any questions.
Okay, awesome. Well, thanks again, and feel free to reach out if anyone does come up with anything.
No, really appreciate the time you put into this, and hopefully you'll get funding and to carry on. And that sounds like I might be getting some specimens to identify.
Yeah, so I was going to say, what are you up to this fall?
Well,
yes, we collected more, not nearly as much as last time, but yes, we have some things, so we'll reach out.
Okay. Right, well, thank you everyone for attending. If there's no more questions, I'll sign off, and thank you again, Heidi.
All right, thank you. Bye.
Bye.